In this video, we will take a look at the slide of eyeball. This slide is stained with hematoxylin-eosin. The wall of eyeball is composed of 3 layers. The first layer is fibrous layer, tunica fibrosa bulbi. This outermost layer in the anterior part of eye is formed by cornea here, and in the posterior part of eye is formed by sclera. It's here, pink layer. The vascular layer of eyeball, tunica vasculosa bulbi, uvea, it's a vascular middle layer which consists of ciliary body and iris and pupil. In the anterior portion eye, and choroid in the posterior part of eye. Internal layer of eyeball, also called tunica interna bulbi or retina. This is a nerve inner layer here. Inside we find lens here, which separate the eyeball into anterior or posterior compartment or segment. Sclera is an opicular layer of dense collagenous connective tissue with numerous elastic fibers and fibroblasts. So this is sclera, and cornea here is a transparent fibrous cap in the anterior part. It is avascular but very sensitive, as it contains significant numbers of free nerve endings. Transition between sclera and cornea is called limbus. It's here. This is transition between cornea and sclera. Anterior epithelium is a non-keratinized stratified squamous epithelium here. The cells in the surface layer have the microvilli and glycocalyx at the apical surface, but not visible in this magnification. Cells of the basal layer are cuboidal and rest on the anterior limiting membrane of Bowman. It's here. It's a Bowman's membrane. Stroma of cornea, or also called substantia propria, contains 60 layers of same thick collagenous lamella and small amounts of keratinocytes. These cells are fibroblasts with branch protrusions. You can see nuclei of these cells. And posterior epithelium is formed with simple squamous epithelium. You can see here. These cells rest on the posterior limiting membrane of the cement. This is also pink eosinophilic thin layer here. These white gaps are artificial artifacts which formed during the histological processing. So this is cornea. Lens is also transparent, biconvex, flexible and avascular structure. The suspension apparatus connects it to the ciliary body, but this apparatus is not visible. Here you can see very thin fibers of suspension apparatus also here. The lens is covered with a capsule of lens. This eosinophilic thin layer. This is a capsule of lens, and it's also transparent layer of collagen fibers and glycosaminoglycan matrix. Beneath the capsule is an epithelium of lens, one layer of cuboidal cells, but only the anterior surface here, but not on the posterior surface of lens. Substance of lens contains the cortex and the nucleus of lens. Cortex of lens contains very elongated concentric hexagonal eosinophilic cells with nucleus and organelles. Here you can see nucleus of these elongated cells in the cortex of lens, and the nucleus of lens contains elements without nuclei or organelles. So here starts the nucleus of lens. This contains remnants of cells without nuclei and organelles. So this is a lens. Choroid is a thin black-brown layer ensuring blood support of the retina. It absorbs the light and prevents reflections. Its basal layer is a Bruch's membrane, basal complex of Bruch, which is a thick eosinophilic layer. It's here. Also here, the Bruch's membrane. The ciliary body here contains smooth muscle, ciliary muscle. Its contraction relaxes the suspension apparatus of the lens and causing its arching and focusing on near objects. Ciliary folds and ciliary processes enlarge the surface of ciliary body and produce aqueous humor. The ciliary epithelium is formed with a double layer epithelium. Their basal laminae are situated on opposite sides due to different embryologic origin of both layers. Outer layer of pigmented cells which continues from the optic part of the retina to the outer layer of the iris epithelium, and inner layer of cuboidal non-pigmented cells is in a direct contact with the aqueous humor. So here it's a inner layer of cuboidal non-pigmented cells. Between these layers, pigmented and non-pigmented cells, is the ciliary canal formed by apical surface of these cells. Here, between these cells, it's a ciliary canal, and the aqueous humor flow from the posterior chamber, from the ciliary canal flow into the posterior chamber and continue to the anterior chamber here, and it is resorbed in Schlemm's canal here. This is the Schlemm's canal, and also in another side. Here is a Schlemm's canal, where the aqueous humor is absorbed. So aqueous humor it's produced between non-pigmented and pigmented cells of ciliary epithelium, and flows from the posterior chamber and to the anterior chamber, and it's absorbed in Schlemm's canal. So iris contains melanocyte. The amount of melanin is in the iris stroma determines the color of eyes. On the anterior surface lacking an epithelium. So here it's not present the epithelium. Aqueous humor in is contact directly with the stroma of iris. Stroma is formed by collagenous fibers and fibroblasts, vessels, melanocytes, macrophages, and myoepithelial cells. There is the sphincter pupil muscle. This is a circular muscle formed by smooth muscle cells, and it has a parasympathetic innervation. In compare with the dilatator papillae, which is formed by myoepithelial cells in fan-like structures and with sympathetic innervation. Epithelium of iris covers only posterior surface of iris and composed of cuboidal pigmented cells here, and the myoepithelial cells are beneath the epithelial cells of iris. So, but it's not very good visible because there are amount of melanin, so it's not clear for description and focusing. The retina is a thin and brittle inner layer of the eyeball. So here is a retina. It functions as a receptor to detect and process light. It consists of 10 layers. The first layer, which is connected with Bruch's membrane of choroid, is a pigmented layer here. This is one layer of cuboidal pigmented cells. This is first layer. Layer of inner and outer segments of photoreceptor cells, rods and cones. It's here. The second layer. Outer limiting layer, adhesive layer between photoreceptor and supporting cells is here. Very thin layer. Outer nuclear layer, the nuclei of photoreceptor cells is here. Very thick layer. External flexiform layer, which contains synapses of axonal processes of photoreceptor cells and dendrites of bipolar cells is here. Inner nuclear layer, nuclei of bipolar cells is here. Inner plexiform layer, synapses of axons of bipolar cells and dendrites of retinal ganglion cells here. Ganglionic layer contains the nuclei of retinal ganglion cells. It's here, very thin layer. Layer of nerve fibers. It's not good visible here. Here you can see the fibers. And inner limiting layer. It's not good visible in these slides. And the lamina separating the retina from the vitreous body. So now you can see yeah, it's very interesting. The light goes through the 10 layers of retina, and where the photoreceptors here detect the light and transform into the signal, and the signal goes from photoreceptor cells into bipolar cells and continue into retinal ganglion cells and go to the optic nerves and into brain. So, and I show you another slide which contains another structure. This is the slide when where we can see optic nerve discs here. This is area without photoreceptor cells where optic nerves exit the eyeball while central retinal vessels enter. So here you can see a branch of retinal vessels, and here is a macula yellow spot area with the highest resolution, contains only cones, bipolar cells, and the retinal ganglional cells, and another layers are reduced. So thank you very much for your attention.